Good morning and good afternoon to all of you for joining our live webinar today, Automated Patch Clamp Assay Development for the Study of Red Blood Cells in Health and Disease. My name is Jason Villagomez, Marketing Manager at Nanion Technologies, and will be your moderator for today's event. Today's presentation is as follows. Joining us as presenters will be Nicoletta Murciano, a PhD candidate working at Nanion Technologies, and Professor Lars Kaestner, Saarland University. Today's presentation is as follows. Lars will provide an introduction into the evidence, prognostic, diagnostics, and therapeutic approaches on RBC function in health and disease. Nicoletta will then proceed and present on her assay development related to channels such as Piezo 1, KCA 3.1, more commonly referred to as Gardos channels, and NMDA receptors, which are critical for CA2 plus regulation used to study healthy and diseased red blood cells. A little bit of due diligence in terms of the audio connection and how to leave a question for us. So your audio broadcast will connect automatically. You can listen through computer speakers, a headset. Also, you can dial in remotely. If you have any questions, we encourage you to ask throughout the course of the presentation, and all questions will be addressed at the end of the program today. Any questions that go unanswered will be reproduced in a transcribed version, as well as a full on-demand copy of the total presentation. If you have any technical difficulties at any point in time, please do type to us and we will try to troubleshoot that for you. Without further ado, I now hand over to Lars for the start of the presentation.
Yeah, hello everybody. So can you see my screen? Is this going well?
We can see you well, we can hear you well.
Okay, so my task today is to present a little bit evidence project, and well, as evidence is an acronym, the total title of the project is a bit lengthy, and it's erythrocytes properties and viability independence of flow and extracellular environment. The project is in principle and a so-called ITN. That means innovative training network. So it's designed to train PhD students, but it also has some well, topical or let's say scientific background. And in the first slide, I want to present you the idea behind the evidence project. And if you look at the investigation of erythrocytes these days, well, one of the typical approaches is the so-called blood smear to investigate, I mean, to smear a drop of blood and then stain the cells and look at this. But then you're actually looking at that cells. Another typical parameter you get out of the blood is the sedimentation rate. What is also a parameter that is measured in stasis. You have several ways of investigations using flow cytometry, for instance, the complete blood count, what is completely artificial conditions. There are transport assays, I mean, membrane transport assays, biochemistry assays, and all this is done or performed on huge cell populations. So what we want to do in evidence is having a closer look, looking at individual living cells and to look at the cells in flow. This is at least the main concept. And to approach this concept, the project has different work packages. And just to give you a very quick overview, so one of the work package is in vivo investigations of blood cells into the circulation. I will present some details of that a little bit later. And at the same time, we try to understand what's happening. So we have a work package on mathematical modeling of blood cells in flow. Then we have more application-like projects, and one is the construction of an artificial spleen using microfluidic approaches. Then we have a huge work package what summarizes all kinds of diagnostic approaches based on red blood cells and also for red blood cells related disease. And then we have a last work package, and also very interesting, and this is about the bioreactor concept to upscale in vitro production of red blood cells. So I want to show two projects a little bit more in detail to give you a flavor of what we are doing. And when I was talking, or when I was mentioning red blood cell flow in vivo, we do this in animal models, in mice or hamsters. Here you see mice, and these mice get such a dorsal skin fold chamber transplanted with a window in it, and this window can then be investigated under a conventional light microscope. And this is how this looks like. What you see here is capillaries, and actually the black objects in the capillaries, these are the red blood cells. And we investigate that under, well, for different disease models. I mean, something like sickle cell disease or neuroacanthocytosis, and we see already differences in the blood flow. So this is a more like basic science project, but we also do something what has more of an application, and we like to transfer things that we see in vivo also in microfluidic assays. And well, here we have a setup where we have like something like an artificial capillary, and you see if the cells flow through this capillary, they have typical, or they appear in typical shapes, and these shapes are, for instance, dependent on the velocity. So what you have just seen is a so-called croissant. So this shape occurs at relative low velocities, and if the velocity of the flow increases, the cells appears more asymmetric. We call this then a slipper. And I mean, these shapes depend obviously on the flow velocity, but also on intrinsic cell properties, something like the membrane stiffness or the viscosity inside the cell, which correlates to the hemoglobin concentration. It depends on the size, on the cytoskeleton, and so on. So with the shapes that appear at different flow velocities, we have a tool to diagnose diseases, to control cell quality, for instance, for transfusions, but also to perform tox assays, because whatever acts on the membrane or the cytoskeleton also changes the red cell shape in the flow. So the changes are sometimes very subtle. So we need a software tool to reveal these differences, and for this we use neural networks for 2D recognition and classification. And what we then get as results are this kind of so-called phase diagrams. So we see under what conditions what cell shapes occur. And this is something like a typical fingerprint for healthy cells, for diseased cells, and all these conditions I mentioned before. So what is Nanion doing in all that? Nanion is a project partner within Evidence, and Jason already mentioned that. Nicoletta, she is a PhD student within this project, and I mean, all PhD students also need an academic supervisor, and that is my role. And obviously, this leads to the topic of Nicoletta's PhD thesis, ion channels in red blood cells. And before I hand over to Nicoletta, I want to mention two points that are maybe important and special for red blood cells in the context of patch clamp. One thing is, and well, this is more or less one of the obvious things, that red blood cells are designed to pass small capillaries. So they are probably among the most complicated cells to patch. Especially if you use the manual patch clamp, because the pipettes you use in the manual patch clamp, they are very much like capillaries. So you need very small pipettes, what results in very high access resistances. So the planar chips in the automated patch clamp are a little bit in favor of patching red blood cells. but still challenging. And the second point I want to mention, what is less obvious, but is also a big challenge, that is the number of ion channels in red blood cells. And I don't mean how many different entities there, I just mean the physical copies of each channel. So I think the best known ion channel in the red blood cell is this Gardos channel. This was also among the first channels found at all. So if you look in the book single channel recordings by Neher and Sackmann, there is a dedicated chapter about red blood cells, and it's about the Gardos channels. But if you look into the cells, like 75% of the cells have less than 20 copies of the channel. And I mean, if you then think of the signal to noise ratio, it appears immediately how a challenge it is to record ion channels in red blood cells. So this, I think, is sufficient of an introduction. So I now hand over to Nicoletta, and she will present you some of her recent data.
Thanks, Lars. Okay, sharing my screen. So can you see my screen?
Yep, can see you well, can see your screen, I can hear you well.
Okay, thanks. So yeah, my presentation will be about the automated patch clamp assay development for the study of red blood cells in health and disease. I would like to start first with a brief introduction on the role of calcium in red blood cells. We know that the calcium concentration is tightly controlled by membrane transport, and this allows to maintain an internal calcium concentration around 30-60 nanomolar against an external concentration of 1.8 millimolar. So any uptake of calcium can regulate many processes, including cell fate. But among these processes, one that is really important is the control of the cell volume and deformability, since red blood cells need to deform to pass through blood vessels constrictions during the flow. But an excessive uptake of calcium can lead also to negative consequences, like the cell shrinkage, promote oxidative stress, and phosphatidyl exposure on the membrane, and the cleavage of cytoskeletal proteins. These accelerate the red blood cells clearance and causes anemia. For this reason, studying calcium regulation is crucial to understand red blood cells diseases. So one of the calcium sensors and one of the first channels found in red blood cells is Gardos channel. Is a calcium activated potassium channel, and this means that uptake of calcium and activation of Gardos results in a potassium and chloride loss, and then an osmotically obliged water loss that lead to cell shrinkage. At the beginning was believed to be a suicide mechanism due to the intracellular calcium increase, but then with the discovery of the first mutation and the associated pathophysiology, the physiological function of Gardos in cellular volume adaptation was doubtless proven. Another important channel involved in calcium regulation is the not selective cation channel Piezo 1. Red blood cells exhibit piezo-dependent calcium influx in response to mechanical stretch. The physiological function of piezo is also involved in the cellular volume adaptation, and there is the interplay with Gardos channel since the activation of piezo increase calcium inside the cell and trigger Gardos activation. Mutation of piezo are associated with the hereditary xerocytosis. And a very important finding was this molecule, Yoda-1, that allowed to activate piezo chemically, and not only so mechanically. Last group of receptors that I investigated so far are the NMDA N-methyl diaspartate receptors. They are ligand-gated, not selective cation channels, showing a tenfold preference for calcium over sodium or potassium. Yeah, as Lars said before, receptors abundance declines during the erythropoiesis, reaching an average of 5 copies in mature red blood cells. And these receptors are correlated with sickle cell disease, since they have abnormally high prevalence and activity in these patients. The channel activation required the binding of glutamate or NMDA and glycine or glycerin simultaneously on the binding site. Amongst the techniques used to investigate membrane transport, patch clamp and in particular automated patch clamp has proven to be a really useful tool. But what changed from manual to automated patch clamp? So the chip replaces the pipette. In this scenario, the chip are pipetted onto the chip, and then the cell is positioned and sealed onto the aperture via suction. And then with a small pulse of pressure, it's possible to get all cell access. The main difficulty in patch clamping red blood cells is the small size of the cells. And our chip are produced to have a really small holes with really high resistance in the range of 8 or 13 milligohm. But which are the features and advantages of output patch clamp? So here we can see our device SynchroPatch that I use for my recordings, and it's possible to record 384 cells in parallel since each well is connected with 384 patch clamp amplifier, and there are 384 channel liquid handling robots that pipette solution simultaneously on all the cells at the same time. So of course, the advantages is that having this high number of cells under the same conditions is very useful to test for the heterogeneity of naive red blood cells conductance amongst cells of the same donor, also between two different donors. And it is a very powerful tool when we want to detect a functional phenotype when we face a new unknown mutation in patient. So the aim of my project would be to develop automated patch clamp assays to study ion channels critical for calcium regulation, and then applies those assays to screen mutated, diseased, or artificial red blood cells populations. But which could be the challenges in studying red blood cells? So as we say, the low number of receptor copies, and first of all, and this is connected to the fact that we are in front of very small currents, and sometimes it's difficult to distinguish from the currents from leak currents. Small size and high deformability that can impact the seal formation. And also there is, it is difficult to discriminate channels that are all involved in calcium transport. So for example, in this scheme, we can see a probable scenario. After mechanical stimulation, piezo is activated. This leads to an increase in intracellular calcium that can activate Gardos channel, and Gardos channel change the membrane potential from minus 10 millivolts to minus 70 millivolts. And this, for example, can lead also to the activation of voltage gated channel like CAF 2.1. So it's not easy to revealing channel identities. Now I will start with a section of the results from my studies. I started with Gardos channel investigation in a healthy donor, and here we can see an example of Gardos experiment. And this is how the panel of 384 wells from our chip looks in our software. Cells are filtered for analysis, applying a quality control on the seal resistance. And you can see in green the wells that are valid for the analysis with a seal resistance higher than 1 gigohm, and the discarded wells in light blue with a seal resistance lower than 1 gigohm. Here is also show the electrophysiological protocol that I applied on cells for studying Gardos, and is a RAM protocol from minus 20 millivolts until to 60 millivolts. And you can see that two cursors in red and in green are placed in this position to the minimum and to the maximum to fit the analysis. So this is how traces looks in our software. These are two graphs where currents are plotted against time. But on the left, we have the time course of the experiment. So these are average current values over time from 19 cells. And the different additions are highlighted in different color. You can see in light gray, the reference addition, in light blue, the activation phase elicited by 50 micromolar of NS309, that is a potentiator of Gardos channel. And then in dark gray, the block by the specific blocker TRAM34 that was used at the concentration of 1 micromolar. So above on the top, you can see currents from the positive voltages, so from the green cursor that I showed before, and below the currents from negative voltages from the red cursors. On the right, we have the same color legends, and here shown the average of row traces. About the criteria that we use for selecting responsive cells, so for Gardos, percentage of activation after the application of NS309 must be higher than 20%, and the block of TRAM have to be higher than the 50%. Point of selected for analysis are shown in this table and in this graph. So we took the last 3 points of the reference addition and the first 3 points of the compound addition and the last 3 points of the blocker. And so from this analysis, then here we have the statistical analysis of currents, absolute currents amongst the different additions with significance between the additions. So this assay that was established in the healthy donor was successfully transferred on a patient sample with unknown mutation of Gardos. Here you can see again the 384 panel wells, and you can see that the success rate was good, close to 65%. And in the green wells are the wells with the seal resistance higher than 1 gigohm. Then here we have 2 representative cells, one from the patient and one from the healthy control. And again, these are current values over time during the different additions. And you can see how in the negative voltages there is an clearly increased in current after the application of NS309. And this is what happened in the control on average. I will not give a lot of details on this because we are still working on the interpretation of data, and maybe this will be planned for a publication soon. Here we have a publication from 2017, and a patient carrying a Gardos mutation was studying, and patch clamp recordings were performed with our device patch liner. You can see on the top some raw current traces from before and after the application of TRAM in the control and in the patient, and then we can see 2 IV curves when TRAM was able to block currents in control and in patient. But when we look at the percentage of block at minus 110 millivolts, the patient cells show an increased percentage of block compared to control, even if it was not statistically significant. But then look at the comparison amongst the standard deviation before and after TRAM. The patient show a clearly increased activity compared to control. So with this study, using a patch clamp technique with our device, it was possible to confirm that the mutation was a gain of function leading to increased channel activity. So here more results from my experiments on healthy donor on the activation of Piezo by Yoda-1. So we have on the left again time course of the experiment with current values over time. And in this case, I show only positive voltages, since it's possible to have a good response compared to the negative, where nothing was observed, at least in this experiment. So the activation was done by 10 micromolar Yoda-1, and the block was done with an unspecific blocker gadolinium chloride. And this is the average values from 13 cells. Same here with the average of raw traces with same color legend. And the electrophysiological protocol that was applied is again a ramp protocol with the minimum at minus 100 millivolts and the maximum at 80 millivolts. And the cursor were placed at the extremes of the ramp. Criteria for the selection of responding cells. So an activation higher than 20% after the application of Yoda is required to select responsive cells. Another publication from 2018 about the characterization of a novel piezo mutant in patient red blood cells was investigated using a synchropatch. And we can see the first column of graphs show raw current traces before Yoda in black and after Yoda in red, in the control on the top and in the patient below. And from the statistical analysis was already clear that there was a difference in the baseline currents. So in the patient, the currents, the baseline currents was higher, significant was higher, and in both voltages. And this difference remained also after the application of Yoda. And we can see that is significant also not only in Yoda responders, but also in the all cells population at both voltages. So with this work, again, was confirmed another gain of function mutation of Piezo 1. So a really nice collaboration with the Professor David Beach from University of Leeds was done recently. And we work on the mechanical stimulation of piezo channels. The Professor David Beach provided egg piezo cell line overexpressing human and mouse piezo and also empty control. And the cells were activated in the same experiment by the fluid shear stress. We simulate the fluid shear stress applying a small amount of solution at high speed at precisely 110 microliters per second, in sync with the triggered recording of the voltage protocol. You can see in these two pictures on the left that we have 16 selected cells that are first activated by only shear stress in human and in mouse cell line. And then we have the combination on the same cells of shear stress plus the activation by Yoda. And we can see how this combination boosts the response in all cells line. Here we have the average from all the cells of the different cell lines, and is clearly evident how the combination among shear stress and Yoda can really boost the response, in particular in the mouse piezo cell line that show a current amplitude higher than the human. The voltage protocol that we used was a continuous recording at minus 80 millivolts, and we placed the two cursors, one for the minimum peak and the other one to calculate the area under the curve. So this assay now is ready, and I'm trying to transfer the assay on red blood cells, but the work is still in progress. About NMDA receptors, I start to investigate this channel in model cells, so in X cells overexpressing the GLU-N2A subunit. with the aim to establish EC50 values for glutamate and IC50 values for the blockers of the receptor. So first I applied a different concentration of glutamate combined to 10 micromolar glycine. And here you can see the raw traces, average of raw traces elicited by each concentrations. And here was possible then to fit a dose response curve that gave an EC50 values of 3.51 micromolar that was in good agreement with those reported in literature. Then I started pharmacological studies applying the different kind of blockers. So MK01, ketamine, and memantine were applied combined to the activation solution with the 10 micromolar glycine and 5 micromolar glutamate, and different increasing concentration from 0 to 10 micromolar from each blocker. Here there is an example from the experiment using MK01, where the black traces are the elicited by glycine and glutamate, and in red we can see traces elicited by blocked by MK801. All the values obtained agreed with those reported in the literature. So yeah, also this assay, the aim is to transfer this assay on red blood cells. I would like to conclude with the current status of the project. So the established assay are the one on Gardos and Piezo channels on red blood cells and NMDA receptors in X cells. About the work in progress, I'm trying to transfer the NMDA receptors assay to red blood cells, and I'm working on the mechanical activation of piezo in red blood cells. I'm also working on the assay development for TRPV2 channels. Future plans would be to investigate NMDA receptors in different stages of erythropoiesis, since different subunits are present in different stages, and also in cultured reticulocytes. So thanks all for listening.
Thank you, Nicoletta. Thank you, Lars, also for the introduction. So now I'll go ahead and moderate some Q&A. And I encourage you to drop us a question if you haven't already done so. So I'll just approach some from Nicoletta's perspective. Nicoletta, how many copies of Piezo 1 are there in the circulating erythrocytes?
So, I mean, in literature, I read some papers where studying proteomics. They estimate a range about few hundred copies per cell, but I think this is a bit optimistic.
And then how do you distinguish between channels involved in calcium transport? You can elaborate a little bit more on this.
Yeah, as I said before, this is a bit challenges in red blood cells, but so mainly we can use very specific activators and blockers of the channel that we would like to test. And we can play also with the concentration, ions concentration in solutions, using precise amount of ions that can help to discriminate currents or to elicit currents with precise amount of ions.
And then another question here. Yeah, sorry, go ahead, Lars.
Yeah, maybe I can add another comment to the first questions of the copy number of channels. I mean, the situation we face with the red blood cells is that there is no translation activity there at all. So that means if red blood cells circulate in the body for an average of 120 days, we still don't know whether the channels that can be detected by proteomics approaches are still functional. So I think the proteomics approaches give us something like a max number per cell, but it doesn't mean it's really that number measurable in the red cells.
Yeah.
And then maybe both of you, since you have some experience here, how does the patch clamp results correlate with the studies compared to using other techniques?
Lars, you want to go first, or?
Well, in principle, one needs to look at this differentially. So I think also already during the early days of patch clamp, like people compared the conductance they got in for particular channels with flux experiments and compared this and try to match it. And I mean, this was a nice way also to get the first ideas about ion channel numbers and so on. On the other hand, I mean, sometimes it's really difficult to how to say this. I mean, if you have ion channels in the cell, the question is what is your gold standard that you say, well, okay, we have this or that channel there. And from my point of view, this would be a functional measurement, and this functional measurement can be some even some amplifying effect, something like to measure the calcium in the red blood cells. I mean, functional measurements are the patch clamp themselves, and I think they are more sensitive than the current methods we have available in the biochemistry approaches. So I think it's you much earlier see a functional recording of a channel than you would detect it in the Western blot. And then if you think that you have this lack of translation in the red cells, there might be a few copies of a channel just as left over from earlier stages, and you can't detect them in the molecular biology methods, but you can functionally see them.
Nicoletta, do you want to add any additional comments? Or
I mean, what I observed reading papers in literature, sometimes for example, the CCP technique is related to patch clamp recordings, even if they are two different techniques, of course, but the relation there I see more in literature, for example. Also calcium imaging can be not perfectly, but can be correlated to patch clamp results.
Also, you teased a little bit, Nicoletta, that you are working on additional research. So I just wanted to make available an upcoming talk that we have, actually part of our 2021 user meeting. So I would provide in the on-demand reproduction of this broadcast sign-up information for your presentation there, which is entitled Mechanical and Chemical Stimulation of Piezo 1 Channels Overexpressed in HEC293 Cells on the Synchrotpatch 34. So I will send the full details of that and where you can read the abstract and also follow alongside the complete program that we have. It's quite a nice lineup. So that's something that you can look forward to in the coming weeks. Also, we would make this available in the next coming days. So stay tuned if you were to join a little bit later on. Once this had already started, we would send it to you in its entirety. If there's no additional questions, then I would stop the broadcast and wish everyone a good rest of their day.